Okay, let's see. Right. So things to focus on. Range size. That's what type of microscopy you use for what. Remember, viruses are mostly electron microscope. Everything else is mostly light. Compared to each other in size, viruses, bacteria, unicellular eukaryotes, multicellular eukaryotes. Microscopes. So that's a repercussion to what we were talking about. So if I ask you a question, which one of the following can be seen mainly with the electron microscope, tell me viruses, right? Branches. Remember those listed? Bacteriology, phycology, mycology. I expect you to match the name of the branch and what it studies. Types of microbes. That same table. It's all questions about that table. So if I ask you, algae are eukaryotic, can be multi or unicellular. Protozoa are eukaryotic, unicellular. Does that make sense? Viruses are acellular, obligate intracellular parasites. Whitaker taxonomic approach based on structure, anatomical details. 5 kingdoms. Know them. Know what each kingdom represents. Like if I ask you the only prokaryotic kingdom, you tell me Monera. Okay. Eukaryotic unicellular kingdom, Protista. Was a Fox classification based on the sequence of 16 sRNA. 3 main groups, Archaea, bacteria, eukarya. Closer to each other, Archaea and eukarya. Binomial nomenclature. Remember the question like Staphylococcus aureus belongs to? Know that stuff. Okay? Ready experiment. It's basically be able to answer basic questions about the description of that experiment. The meat and maggots experiments. Louis Pasteur, I refer to that experiment, famous experiment with a curved neck flask. Know the contribution made by Semmelweis. First, the hand wash to curb the puerperal fever in the maternal ward. Joseph Lister, invention of antiseptic surgical technique using carbolic acid. Koch's postulates. You isolate microbes from the sick but not healthy organism. Isolated microorganism can be grown in a pure culture. It causes the same disease in the experimental infection, and you isolate all the same organism from the experimental animals or humans succumbed to the infection. Microbes are ubiquitous. Now, for importance of microbes in nature and disease-causing agents, very, very simple ideas. Basically, like, which of the following diseases is not caused by microorganisms? Like, diabetes is not, okay? Fracture is not caused by microorganisms. Industry, food, drug production. I'm not going to ask you specific examples. It's multiple choice. Okay, just know that they play a role. That's basically it. Clostridium acetobutylicum can produce fuel. Understanding changes of wine microbiome. You can control the quality of wine. Now, for this one, how microbes can themselves be a source of antibiotic. Remember, antibiotics are the chemical warfare because they fight other microbes. So this is how microbes produce antibiotics. So far, I know I'm going fairly fast. It's all getting recorded. It's all going to be on YouTube. It's all going to be in recordings on the blackboard. Yes. Clostridium acet. It produces fuel. Okay. So gram staining for Clostridia, positive. Function of endospores, protection of DNA. Clostridium botulinum causes botulism. How does it do it? Basic mechanism. inhibition of acetylcholine release at the neuromuscular junction. Antibiotics, would they help? No, because it's toxin. Transmitted mainly foodborne. Clostridium tetani causes tetanus. I'm not going to write the name down. How does it do it? It inhibits the inhibitory relaxation neurons. Spastic paralysis. If you don't mind, I'm going to see it. It's going to be easier for me. Okay, transmission, environment, antibiotics don't help. Clostridium perfringens, gas gangrene. Antibiotics, limited help. Transmitted environment. Clostridium difficile, C. diff, antibiotics, limited efficiency, transmitted orally. Superinfection is caused by depletion of good microbes. Treatment options: fecal transplant. Nosocomial means hospital acquired. Hydrogen bonds. I want to make it super clear since I screwed up on the slide. Hydrogen bond is between partially negative oxygen and partially positive hydrogen. Cool? I don't take pluses and minuses as complete charges. I'm too lazy to write little delta. pH, concentration of hydrogen ions. All you need to know about this: low pH is acidic, high pH is basic, 7 is neutral. Carbohydrates made of monosaccharides. The general formula, CH2ON. So like glucose is going to be, N is going to be 6, for instance. You don't need to know the general formula. Know that there are carbon, hydrogen, and oxygen. Polysaccharides are formed from individual monosaccharides. Can be broken down by addition of water. Synthesis is reaction of dehydration. Functions, mainly energy, some structural. Triglycerides or fats made of glycerol and free fatty acids. Reaction, dehydration. Functions, energy. Phospholipids, I'm not going to draw the structure, but you have phosphate, glycerol, and free fatty acids. Functions, the membrane, main structural feature, they are amphiphilic. No. Meaning they have hydrophilic and hydrophobic parts. Sterols and hopanoids strengthen the membranes. Sterols in eukaryotes, hopanoids in bacteria. Proteins, know that they are made of amino acids, dehydration, reaction of dehydration. Functions, basically everything in the cell. Structural levels, from primary to quaternary, I want you to be able to match a description. So if I mention alpha helices, you tell me secondary. If I mention sequence of amino acids, polypeptide chain, primary. Okay, several individual subunits together, quaternary. Denaturation, loss of structure because of some kind of an insult. thermochemical function suffers as well. Nucleic acids, DNA, RNA, made of nucleotides. Functions, storage and expression of genetic information. So nucleotides, just, that idea, you have a base, sugar, phosphate. Between DNA and RNA, structurally, DNA is double-stranded, RNA is single-stranded. DNA stores genetic information, RNA helps with expression of it. Complementarity, ATGC, that's for DNA, or AUGC, that's for RNA. Genome, all the genes in the organism. Gene encodes a single polypeptide or protein. Genotype, well, basically, a certain arrangement of genes in the genome. Phenotype, physical appearance. Remember that gene expression, DNA, RNA, protein, defines phenotype and its gene expression at the given moment. Mutations are changes in the DNA, and they greatly contribute to adaptation and evolution, since mutations can lead to different phenotype, which can be more fit for certain selective pressure. Main shapes: cocci, bacilli, spirillum, spirochete. I give you a description, you tell me what it is. Make sense? 4 parts: nucleoid, membrane, cytoplasm, ribosome. Know the functions. Understand osmosis, tonicity, what happens to the cells in this hypertonic and hypotonic environments. Am I clear with this? I will ask you questions. Cell that is placed in, I don't know, hypertonic environment will blah, 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 blah. Okay? Know how cells can survive in conditions with high osmolality. We mentioned it. They maintain high osmolality within the cell. Cool? Okay. Transport of nutrients. Know the difference between diffusion and active transport. In terms of diffusion, know the difference between simple and facilitated. So simple, directly through the membrane. Facilitated, you have some kind of transmembrane protein. Active transport requires ATP. Okay. Cell membrane consists of phospholipids. There are transmembrane proteins. Now, know the function of the membrane, like, transport, basically. Function of hopanoids, strengthening the bacterial membrane. Transmembrane proteins, transport and signaling. Inclusions, remember there are no membrane-bound organelles in the bacterial cell. Inclusions are not membrane-bound, they store things. Endospores protect DNA. Now understand, you have to know what triggers sporulation and germination. So if I tell you dry, no nutrients, you tell me that will stimulate sporulation. Wet, a lot of nutrients, will go back to germination. So know that this processes, they go like this. So sporulation, so spore, okay, that's a poor representation. Spore, vegetative cell. Okay? So when spore goes to vegetative, that's germination. But vegetative cell goes into the form of endospore, that's sporulation. cell wall. Now, got a question about, you have to be able to distinguish first and foremost gram-positives, gram-negatives. So gram-positives, cytoplasmic membrane, thick wall. Wall contains teichoic acids, which are virulence factors. Gram-negatives, cytoplasmic membrane, thin cell wall, outer membrane. Outer membrane contains lipopolysaccharides and porins. Lipopolysaccharides are virulence factors. Porins facilitate transport. Polymer, peptidoglycan, consists of carbohydrate chains connected by the peptide linkages. Antibacterial drug can inhibit the formation of peptide linkages, essentially ruining the wall. Outer membrane and cell wall is separated by the periplasmic space. Lipopolysaccharide, just discussed, it causes general inflammation. Okay? It's a virulence factor that causes general sickness. Now, for mycobacteria, they are technically gram-positive, but mycolic acid in the outside of that gram-positive envelope. So, type of staining that we use to identify them, acid, fast. Do every bacteria have cell wall? No. No, Mycoplasma does not. Don't mix up Mycoplasma and Mycobacteria. The only difference that you need to know between archaeal and bacterial cells is that archaea do not have peptidoglycan. They just have different molecules. Glycocalyx. Question? Oh, okay. So, archaea do not have peptidoglycan. They have other molecules like proteins and carbohydrates. Okay. Glycocalyx, you need to know capsule slime layer. Capsule increases adhesion, protects from phagocytosis. Slime layer only increases adhesion. So, this is what I'm here basically asking. So when they increase adhesion, either slime layer or capsule, they facilitate the formation of biofilms. Bacteria in the biofilms are better protected against antibacterial agents, immune responses from the host. So biofilms are more resistant to drying out. They are more sustainable. This is why biofilms, much harder, that's the clinical importance, to get rid of. They're harder to destroy. Know the difference between these three. Functional difference. 4 types of flagellar arrangement. That Peritrichus, Lophotrichus. Now, moving using flagellum. Know about run and tumble. And energy, diffusion of hydrogen ions through a special channel. So far, we're clear. Chemotaxis, movement, positive chemotaxis, movement towards the increasing concentration of chemoattractant. Negative is moving away. Gram stain for staphylococci, positive, main skin diseases. So, remember that skin diseases, know them. Pythagoras called it skin, furuncle, carbuncle, necrotizing fasciitis, all that kind of stuff. I'm going to ask some of those. Now, you don't need to know this too. Exfoliative toxin breaks down the basement membrane, contributing to cutaneous diseases caused by Staph aureus. We didn't talk about beta-lactamase, so don't worry about it. Subcutaneous tissue, necrotizing fasciitis. Toxic shock syndrome toxins. So what I want you to know about this one. TSST hyperactivates immune system. Okay? And leads to toxic shock. Other conditions for Staph aureus. sinusitis, osteomyelitis, endocarditis. Can it be a part of microbiome? Yes, usually in the respiratory tract. Transmission route, contact, and respiratory. Look, when I'm telling you something here, if I tell you you don't have to worry about it, you don't have to worry about it. If I tell you transmission routes, I mean it. You need to know these two transmission routes, and they're there, contact and respiratory. Staph saprophyticus, urinary tract infection. Transmission route, either sexual or via microbiome. Don't mix up Staph aureus and Staph saprophyticus. Staph aureus is not sexually transmitted, thank God. Staph saprophyticus is. Staph epidermidis, normal skin microorganisms, so yes, it's a component of microbiome. Can it be pathogenic? It can be in the immunocompromised patients. Now, I'm going to skip a huge chunk here, just telling you. Remember we talked about parts of eukaryotic cell. Only the parts, the organelles that we talked about, I have a right to ask about those that we talked about, that are in the PowerPoint. I give you a function, you tell me an organelle. Does that make sense? Or vice versa. The function of, I don't know, mitochondria is, and you lay it out. Am I clear? A couple of things to know. For 3 filaments, when asked about the features and functions, not so much, but features you have to be well aware what they're made of and whether they are dynamic or static. So if I ask you about microfilaments, you need to know they're made of actin. They can be destroyed and rebuilt. Endosymbiosis. That whole idea about mitochondria and chloroplasts emerging as the result of endosymbiosis. Am I clear? Structure and functions, basic stuff like cresta is mitochondrial, thylakoid is chloroplast. Okay. Know that they both have DNA and ribosomes. Okay. So they do have DNA and ribosomes. Other structures similar to mitochondria are hydrogenosomes, kinetoplasts. Extracellular matrix, increased adhesion, protection against phagocytosis, all the same story. Flagella and cilia, again, just know the functions. Protozoa. Remember classification of 4 classes. Mastigophora, Ciliophora, Sarcodina or amoeba, and Apicomplexa. Match the description with the name. Structural features, they don't have cell wall. Disease, Naegleria fowleri, brain-eating amoeba, transmitted from the water, goes into the brain. It's not a communicable disease. You cannot transmit it to another person. It's confined to your brain. Prognosis is poor. Treatment is futile. Infection is infrequent. Am I clear? Fungi. Look, I'm not going to ask you about reproductive approach. Relax. Zygomycota, basidiomycota, ascomycota. I'm not going to ask this. I will ask about this. Molds, yeasts, and dimorphic fungi. So you have to know, like yeast, single-celled, molds, multi-cell, dimorphic can be both. Structural features like hyphae. I'm not going to. Spores are reproductive structures. Histoplasma capsulatum is dimorphic. It's mold in the environment, yeast in the human body, transmitted respiratory route. Animals are involved. This disease is zoonotic. It is not communicable between humans. Animals that are involved are birds and bats. Histoplasma is found in their feces. Causes histoplasmosis, mostly lung infection. Okay? Respiratory form is treatable. Systemic is hardly treatable. It's rare. Part of the S that is affected the most, I would call it Eastern Midwest, if I can do this, like Kansas, Nebraska, Ohio, Indiana, Illinois, Midwest. Helminths. Classification, 3 groups, nematodes, cestodes, trematodes. Know the shape, know the sex. Segmentation, body cavity. That's that. Tinea solium. Know the life cycle. So if I ask you what do we eat, you tell me we eat larvae. Okay? So we eat larvae. We shed eggs. Larvae after developing, so eggs develop into larvae in the intestines of the pigs. Larva migrates to the pig meat. Scolex is the head. Suckers used to attach to the surface of intestine. Proglottids, small elements of that carry eggs. Normally, adult worm lives in the gut, feeds by diffusion. Infestation is asymptomatic. Cysticercosis, somebody consumes eggs. Eggs develop into larvae. Larvae migrate into human tissues and commonly infest muscles and brain. Pathological effects of cysticercosis: if it's in the muscles, it's myositis. If it's in the brain, it is encephalitis, inflammation of the brain. Treatment: all you need to know is that there is one. For prevention, cook your meat thoroughly. Viruses. You need to know the parts like for naked viruses, it's a capsid and genome. For envelope capsid and genome. Does that make sense? What kinds of genomes do they have? They have RNA or DNA, single-stranded or double-stranded. They're not alive. Papillomaviruses, double-stranded DNA, naked. So they are non-enveloped viruses. Major diseases: warts, genital and skin warts, unfortunately cancer. They infect basal cells of the stratified squamous epithelium, these viruses. They do not require cancer for the replication, but they cause cancer by increasing proliferation, cellular division. Cells accumulate mutations and eventually turn into the cancerous phenotype. Gardasil is the vaccine that protects against 2 strains causing cervical cancer. Light microscope. Now, light, compound, binocular, parfocal. Know what each of these 4 terms means. Does that make sense? Refraction, changing the direction of light. Reflection, well, it's reflection. Know how to calculate magnification. Okay? Magnification of the objective multiplied by the magnification of the eyepiece. Resolution, ability to distinguish small details. Contrast, color difference between the sample and the background. That's how can we determine, just talked about it. What limits the resolution of the microscope? The wavelength. Who invented microscopy? Anthony van Leeuwenhoek. Major parts. Okay, now this is sort of a review for you. Like if I ask you, what is the function of, I don't know, iris diaphragm? Regulate amount of light that falls on the specimen. It's probably the only one that is kind of hard. Okay. Major types of light microscopy. You see I left you a lot of space here. 7 types. Match the name and the description. We observe cells against the lit background. That's bright film. Okay. 2 types of electron microscopy, transmission and scanning. Again, match, description, like if I want to see the surface, I'm going to use scanning. Fixation, making the specimen dead and unmovable. So it doesn't move anymore. We stain specimens to increase contrast. Simple stains. Know the difference between positive and negative stains. Positive stains stain microbe. because they positively charged and bind to the negatively charged microbial surface. Negative stains stay in the background. So that's what I'm referring to here. Okay. Differential stains. 5 types. Obviously, I'm going to ask only about 2. Acid-fast and gram stain. So, step-by-step procedure. You don't have to recite it, but I may ask you, what is the role of alcohol? It's decolorizer. What is the role of iodine? It's mordant. Bacilli. Positive. Common virulence factor. Endospore. Bacillus cereus, food poisoning. It's not an infection, so it's intoxication. It's caused by the bacterial toxin. Bacillus thuringiensis, used for production of insecticide. BT. Bacillus subtilis, research object. Bacillus anthracis, causes anthrax. Forms. Cutaneous, enteric, respiratory. Respiratory is the most severe. Transmitted, mostly by contact, in rare cases with food or with inhalation. It is zoonotic. Reservoir is cattle or other hoofed animals. 5 steps of microbial cultivation: inoculation, incubation, isolation, inspection, identification. Physical state. I give you a description, a thorough description of media. You have to be able to tell me if it's solid, semi-solid, or liquid. Okay, if I tell you it doesn't have solid surface, you tell me that's semi-solid. Cloth-like. Composition. Know the difference between defined versus complex. Okay, defined or synthetic versus complex. Does that make sense? So if I tell you like chicken broth, that's complex. Purpose. General, enriched, selective, differential. Again, description, Types of cultures. Pure, mixed, contaminated. We use cells for virus cultivation. Which live organism? Look, I'm not going to ask you, can we use elephants? Yes, we can. It's too expensive, but we can use elephants to cultivate microorganisms. Basic limitations: it's expensive. They have to be free of any other pathogens. Okay. Incubation happens to control for the environment, to increase reproducibility of results and to compare results between different specialists. Main isolation techniques: strict plate, pour plate, spread plate. Note them. There will be a question. I will describe one of these methods and you will have to identify which one I am describing. Strict, pour, or spread plate. Isolation to separate different species. Culture inspection. Microscopy, visual inspection, color, shape, blah, blah, blah. Now, this is what we were talking about today. Don't be afraid of these questions. I give you a description. Let's say I tell you we use fluorescently labeled antibodies to detect genes, to detect antigen on the surface of the cell. Okay, fluorescent labeled antibodies, surface of the cell, immunofluorescence. We identify a unique DNA fragment that belongs to certain species using primers, okay, and amplification, PCR. We use laser beam to ionize a fragment and run it, and separate them based on mass to charge ratio. Mass spec, MALDI-TOF, okay? Does that make sense? This is going to be, I promise, this is going to be the questions that you're going to see, okay? We identify the antigen that is captured by the antibody on the plate, ELISA, okay? We separate proteins first and then identify the protein of interest using antibodies, Western blot. Make sense? Have a general idea what's going on so you can recognize the description. Am I clear? So we separate proteins in the sample and then identify the protein of interest using antibodies. It's Western blot.